Thank you. Thank you so much for the invitation. And I'm so sorry I couldn't come. I actually was an undergraduate exchange student in Nottingham. I would have loved to come. And anyway, the CCP-EM community is amazing. So it would have been great to be there, but I'm actually pretty honored and happy to be here nonetheless. Okay, so I guess my lab mostly uses cryo-electron tomography, and I like showing this slide just to say electrons are the best radiation that we have to look at biomolecules, and it really shouldn't matter whether this is inside and outside of cells. So it's exciting to think about cryo-EM as a high-resolution tool for cell biology proper, not only structural biology in situ, but really biological discovery as well. And my lab, I like to say that I'm from Mexico City, and my lab has grown in similar directions as Mexico City grew, so pretty random. But today I want to talk to you about this part of the lab, which is bacterial cell biology, which we found very satisfyingly that cryo-electron microscopy is a useful tool to discover new biology. And the dirty secret that we sometimes don't tell our friends is that when we take a tomogram in a mammalian cell after milling and stuff, you're covering a really tiny proportion of the cell. And although I am sure, and I apologize because I haven't seen, but I'm sure that there's beautiful talks that you've seen using cryo-electron tomography, we really need to plan the experiments very, very well in order to go to the right place, get enough statistics, and unveil interesting events. Whereas bacteria has a little bit of the opposite problem, where in mammalian cell biology, fluorescence microscopy gives these stunning images where you can really localize, in subcellular compartments and things like that. Bacteria are very, very small. And they're really just one order of magnitude larger than the diffraction limit of light. And therefore, doing bacterial cell biology live imaging is typically kind of difficult. Conversely, if we make lamella out of bacteria with cryo-electron tomography, we can get up to sort of like about half of the cell included in our lamellae, and then typically you have dozens of cells or per lamella, and then probably a couple of hundred cells per experiment. So that really allows us to be more likely to find events of interest and to observe sort of like different states of the bacteria. And so today I want to tell you a really fun story that's been unveiling in my lab for quite some years now, pretty much since I started my lab. And it's related to a class of phage that are called jumbo phage. And so everybody knows phage. Everybody who likes EM knows phage really well, but everybody pretty much in the world, there's like a lot of molecular biology tools and a lot of biology discovered through phage that have been fighting bacteria for billions of years. And phage have about 30 to 180 or so kilobase pairs. Here's lambda at about 50 kilobase pairs, this sort of ubiquitous, wonderful phage that everybody knows and loves. And T4, which is supposed to be or thought to be a pretty large phage, is at about 150 kilobase pairs. Now, recently it's been unveiled that there's this new class of phage that are called jumbo phage, simply because they're huge. They have really large genomes from 200 to 750 kilobase pairs, and they're just really large. And when we started working on this, people were wondering, well, are these exotic, random, did you just find a very exotic organism? And don't get me wrong, I really like exotica, but this was more related to these phages were not found simply because the filters that people used to look for jumbo phage were just too small, 100 to 150 nanometer filters. Here we're talking about phages that are 200 nanometers or so, simply weren't discovered because they just were not included in how we acquired the sample. But these phages only here in San Diego at UCSD have been found in the gut of newborn babies, sea spray, aerosol sea spray, cheese, you name poop, a lot of poop. So you name it, wherever you think that phages should exist, or really in any area, anywhere that there's an organism or a bacteria, you're going to find this jumbo phage. So very exciting. I started working on this phage because my dear friend Joe Pugliano is a microbiologist, and he started looking through these genomes about 10 or 12 years ago. And then he found that among other curious and weird things in the genome, there was one genome that seemed to be a homolog for tubulin. And so the way that the cycle of these phages works is that you have the phage gets infected in the bacterium, the bacterial genome gets immediately degraded, And then when Joe recently tagged these tubulin-like analogs called fuzz-Z, it will basically start forming filaments just like microtubules at the edge of the cell. These microtubules seem to catch, or fuzz-Z filaments seem to catch a ball of replicating phage genomes, centered, looking a lot like a mitotic spindle. And when there were enough phage in the cell would lyse and go on to infect different cells. So Joe collaborated time with Dave Agard, where they purified this protein, solved the structure, and then basically looked like it really looks like a tubulin, smells like a tubulin, and acts like a tubulin. It has dynamic instability. It can treadmill in vitro. So everything that you would imagine from a microtubule fuzz-Z has. So when I came to UCSD, which is already like 8 years ago, Joe was thinking at, okay, well, it's this really large genome. If there's a tubulin in it that seems to form something that looks like a spindle, what other interesting proteins are there that are not proteins that make phage? Because you have to obviously make a lot of capsids, a lot of tails, a lot of plates and everything. So like to put the phage out into the world, but what else is going on? And the protein that was most expressed was a protein called GP105. That's gene product 105, not glycoprotein 105. From a phage that infected, a phage called FIC-AC that infected Pseudomonas aeruginosa. All right, so this is what the fuzz-Z filaments look like here in green fuzz-Z, and in blue it's DAPI. You can imagine this dotted is the cell. And then when Joe fluorescently tagged this other protein, GP105, it seemed to localize to the periphery of the replicating genome. So here you see the cell in red, and then blue for DAPI, and then GP105 around the genome. And they're like, oh my God, it has like a spindle. And now it looks a little bit, it looks a lot like a nucleus of a eukaryotic cell. So I'm a physicist by training. I thought most likely this is a diffusion reaction problem. So this protein probably binds DNA, diffuses in there, and gets stuck. And that's the end of the story. But this is the best and not very satisfying way that we could see this at the time. And I'm sure you guys have had a problem where sometimes your friends talk to you about their science and you're just very excited for them. But sometimes it's really sort of like deposits in your head and itch that you have to scratch, right? And I just really needed to see with this protein work about it. People in my lab were very excited. So enter the epic Kanika. Kanika was the first student in my lab. She was joined with not only Joe, but Kit Pogliano, and she really kind of knocked that out of the park. And the first thing the Kanika work did, along with Felix Wagner, who was a master's student in my lab at the time, is to figure out how to make monolayers of bacteria so that she could make lamellae. And so, to my knowledge, this is the first time that we made bacteria lamella from bacteria. And for those in the audience trying to do that, it is actually hard. It takes a lot of optimization. So please hang on and keep trying. Each new bacterial species that we use takes a little bit of work. Anyway, so you can see over here that Kanika has a lot of different bacteria, and some of them will have been infected by phage. And so when we take a look at one of these, this is one of the first tomograms. This is already quite some time ago that Kanika took a look at. And the first thing that was stunning is that, contrary to what I thought, there is actually a shell of DNA. There was also this controversy that we were thinking, is it possible that there's a membrane in there, sort of like a nucleus? And membrane dyes are very hard to get into bacteria. So here you can see that there's no membrane, or you'll see in a moment, there's no membrane. There's a hard shell that seems to be one protein deep. So basically a meshwork, a 2D meshwork made out of protein. And then something we didn't know was very cool to see is that the capsid seem to be attached to this thing. So you can see here a capsid that's basically more or less empty, a capsid that's more full. And then at some point, these capsids seem to graduate as they get enough of all their genome inside and sort of come and get a tail and so forth. The other thing that I want to point your attention to, other than this, protein being sort of like a single layer of protein, is that the ribosomes, you just see here segmented in yellow, are actually completely excluded. So I'm telling you, the genome is outside of this it's inside of this compartment, and the ribosomes are outside. And the landmark of a eukaryotic nucleus is that it separates DNA replication and transcription from translation, right? So if this operates as a nucleus, that means that mRNA has to be done inside this compartment and exported so that the ribosomes translate it. But it also means that proteins need to be imported into this thing. So that's a little crazy. So to test that hypothesis, like, is this thing really acting like a eukaryotic nucleus? what we do is in Joe's lab is to uninfect, in the uninfected cells, we fluorescently express a protein that we want to test what its localization after infection. So here is GP237. This is a REC-A-like protein, therefore it should be inside the nucleus. And as you can see, when it's infected, basically the whole protein, REC-A, as you should know, should bind to DNA, and it completely gets absorbed or sort of like imported into this nucleus compartment. And it's the same thing for a GP107, which is a subunit of a very sophisticated phage polymerase, also gets completely imported. And then if you look at topo, so from the host, so any protein that we test that you would imagine would be inside the nucleus, whether it came from the host or from the phage, would get imported into the nucleus. And conversely, ribosomal proteins we already knew from tomography, but they get when infection happens, they completely get excluded. So they stay in the so-called cytoplasm, or as Tom likes to call it, the extracellular domain. And then even kinases. So any metabolic or translational related proteins that we would test stay excluded. So this really tells you that there has to be control of import and export from this compartment, and that it really functions like a eukaryotic nucleus. And if you didn't think everything that I just told you was a little crazy, I want to show you the entire structure rotates. So as the filaments bring it into the center of the cell, it starts rotating round and around. You see like an entire view, which if you think about it, it just seems like a really large thermodynamic cost of dragging a large object around and around inside the cell. All right, so we were curious about how the virions find the nucleus. I'm going to go a little bit quick through that part because I'm mindful of the time. But the capsids assemble at the inner membrane. Somehow they find this nucleus. They get their inner body, all the DNA injected, and eventually they graduate. But the question is, how do they get into the nucleus? How do they find the nucleus? And so this is fluorescent microscopy. Now what you're seeing is GP105, the shell protein, shown in red, and the capsids shown in green. And I'm going to show you a movie on the right, although again, bacterial cell biology is really hard to see. But if you were able to see this, what you would see is that the proteins, they seem to be kind of like walking in a line from the pole of the cell into the center until they reach the nucleus. And so, of course, this got us to think back about fuzzy, the filaments. And so now here you see fuzzy in red and the capsids in green. And I'm going to show it to you in the right because I think it's hard to see. And what we saw is actually that the capsids are co-localized with the filaments, which is a little crazy because I already told you that there's a nucleus and there's microtubules, and I'm telling you that there's microtubule transport. And so we wanted to know how this was doing. We obviously looked for tubulin binding domains in the genomes of these things, and we couldn't find any. So we decided to take a look directly with tomography, again, Konica, and this was not trivial because there's only a couple of filaments per cell, or at least on each side of the cell. And so Konica and Vorapon in Joe's lab made a mutant of fuzzy that basically cannot hydrolyze GTP. So you have these very, very long filaments, and really cool to see the virions or the capsids gets like completely stuck along the filaments before they reach the nucleus because there's no GTP. All right, so that made us think, so this is a tomogram that Konica took. Well, you can see that she captures a couple of filaments and a lot of empty capsids along the way. And if you sort of like, I'll point your attention to the image on the lower right, and so where the filament is and the capsid, and what it really looks to be happening, it's a direct connection. It's only about 4 nanometers in distance, sort of about the same distance that that you see sort of the capsid connected to the inner membrane that I showed you before. And it seems to have the capsid connected from a vertex directly into the filament. And so this and the fact that we didn't find any tubules got us to think that maybe the capsid is attached directly to the microtubule or to the filament fuzzy, and then it's the filament that is moving around. So in order to test this, we decided to use a method called FRAP. Again, it's really hard to use FRAP in this tiny, tiny bacteria, but FRAP is fluorescent recovery after photobleaching. So what you're going to see is a bleach in the fluorescence of fuzzy, and then we're going to try to follow whether the fluorescence moves along. This would mean that the filament itself is treadmilling, so it's advancing, polymerizing from one side and advancing to the other, or whether the the filament is static, and then there must be another way to transport the microtubules that we needed to find. So here's FRAP. I'm going to show you the movie, which again, it's very hard to see for bacterial cell biology. And so I'm just going to show you the snapshots or the stills. And you can see here that the region that we FRAP basically starts advancing towards the center of the cell. And so what that means is that new fussy subunits are added on the back, and that sort of like pushes the subunit in this region, that in this case the bleach part towards the center of the cell, and that means that the filament is treadmilling, as again Dave Agar's lab had seen before in vitro. So basically, I told you that the capsids or the filaments treadmill, that the capsids migrate along these filaments, and that the phage nucleus rotates. And they all do, and basically they all do it effectively at the same speed. And so our working model is that subunits are added at the pole that basically pushes the filaments or the capsids along towards the center. And then when the filament hits the nucleus, what we think is like it hits it, exerting a force that makes it rotate. The filament itself, the nucleus rotates, the filament depolymerizes, and that allows us to deliver the capsid into the shell. And the rotation, we think, is because you can't diffuse in 2D in a protein shell. You can do it in a membrane, but not in a protein shell. So this rotation is an ability then to clear the space, like clear the real estate for new capsids to come into this thing. Okay, so but what I haven't told you all this part is that what the function of this nucleus is, and it's an anti-immune defense compartment. So some phage chemically modified DNA, this phage just builds a wall, it builds a room, and it says, you know, I'm going to keep my genome inside and you can't enter, right? And so CRISPR cannot come in, and 2 different labs have shown that, that there's not a lot of things that could come in unless they want to, as we showed you before. So the question is, so this is, by the way, when Tom Laughlin, who's like amazing Tom, came to my lab, and he wanted to know how does the nucleus assemble without using seal? And he was assisted by the magical Sergei Suslov, who was in my lab and is now an app specialist for thermo that many of you have probably met. So the question that Tom have is like, how do you have a protein that grows from 200 nanometers, or sorry, like a compartment? So this is the initial infection, which is about 200 nanometers in size. So this is one genome. And then how do you do that and grow like 10 times its size by adding protein all the time, and all the time not losing its seal? How do you keep the nucleases out, but some protein seen, and so forth? And so here in pink on the top, you can see this little beads on the string. That's a side view of the compartment. So like, again, a single compartment. And here at the bottom, you can see, a top view that Tom got of this thing that looks sort of like a little meshwork. So Tom wanted to get the structure of this thing, and he started using subtomogram analysis. When Tom came to my lab, he told me, when you showed me the images of the phage nucleus, it looked really round, and now it turns out it's like incredibly flexible. It'll, you know, bend all to like minus 30 degrees to 90 degrees in the other direction. And so Tom took little patches, sort of like oversampled the surface, obviously used ribosomes as fiducials. You guys know the story. And then basically got a subtomogram average that looked like a waffle. And what was interesting about this subtomogram average is that Tom, he checked this 5 times because he didn't believe it, is that it seems to be formed by tetramers. So this protein makes tetramers, and then these tetramers assemble into sheets. And so this tetramer, you can see here in red, looks like a pinwheel, and it has obviously a center fourfold and a corner fourfold in it. And so at this point, we decided that we should name the protein. This protein I told you was called GP105, but in the different phage, that protein will be GP53. And I'll just show you a little bit more about genomes in a second. So we decided to call the protein, and my friend Kevin Corbet, who I'll talk about today, decided that there's probably enough Greek and Latin names out there. So we called it Chimalin after the Chimali, which is an Aztec shield that warriors would bring into battle. So since this whole compartment is shielding the genome, we call it Chimalin. So I'll call it this from now on. All right, so I told you that Chimalin was a tetramer, and we are all used to tetramers in proteins. That's not new, and it's actually a very ubiquitous multimeric assembly. However, for building things, nature doesn't like tetramers. Nature typically likes hexagons, and as you know, with the occasional pentamer at all sorts of scales. This is not the preferred tile. We did find, from what Tom called an exotic bacillus, an S layer, but of course S layers are embedded in membranes, and therefore it's easy to sort of like fill in the gaps. So Tom figured out that because of the curvature of this thing, it was subtomogram averaging wasn't being very simple. And anyway, at some point we thought it would be easier to ask my friend Kevin Corbet, who's a fantastic structural biologist, who convinced his postdoc Amar to see if he could crystallize chimalin. And so Amar had a very nice segmental data that you can see here on the upper left, and the monomer on the right, it was a clean peak, and he tried and failed to try to crystallize it. So Tom asked Amar, well, let's take a look at the void peak, here's the B peak on the left. And it was very cute to see that it is very, very high concentrations. Chimalin will actually form little closed compartments, but then we came back to the same issue of the curvature. But so Tom started to try to analyze it, but for a good measure, he also threw this peak, that's the C peak, the one in the middle, that corresponded to about 24 subunits of the 70 kilodalton protein chimalin. And I told you that this thing forms tetramers or squares. And so the smallest closed compartment that you can make from squares is a cube, which is exactly 24 subunits. And super excitingly, what they had on their hands are actually cubes of chimalin, squishy cubes, not a perfect assembly, but squishy cubes. And so Tom decided to give it a shot at this with using good old single particle cryo-EM. All right, so here, oh God, I'm so sorry. I'm gonna have to do one thing very fast. I made something bad with this movie yesterday. Okay. Ooh. There you go. All right, so here's the structure that Tom got for chimalin originally. As you can see, it's a tetramer. It's sort of like a cube of these little squares. And then Tom did expansion symmetry on the tetramer. So I told you it's sort of like a rather squishy thing. And then he did the same for the monomer there on the left. And he did this for two different species. One from phage that infects E. coli and one from a phage that infects Pseudomonas. And his highest resolution structure, I think it was about 3.1 angstroms. And I have to tell you that the only homology that we had found to this thing in terms of structure was actually a tRNA acetylase, which of course makes no sense to build things from a toxin antitoxin system. So here's the structure of chimalin. It has a core protomer where about 2/3 of it has this fold similar to a tRNA acetylase. But the really interesting bit about it was that it has what we call invading termini, where the N terminus sort of goes to invade or hug, like I like to say, another monomer within the tetramer, sort of like, and this is how the tetramer gets assembled. But then the C terminus actually does the same, but not for only one, but two neighboring tetramers, as you can see here in the threefold corner that you have in the cube. And then Tom showed that the rigid body molecule of the C terminus actually allows you to go into the flat surface. So this incredible variability and flexibility and curvature is possible with these two domains that really even allows you to go from a threefold cube corner to a fourfold in a sheet. So Tom assembled the sheet of the tetramer, and we wanted to know, you can see that there's some holes in here, and we wanted to know if these holes would be sufficient to make new chimalin subunits. I'm sorry, to export and import proteins. And my friend Romy Amara at UCSD and her fantastic student Christian Seitz went out to test that using molecular dynamic simulations. And as you can see from the simulations, basically the pores actually have some diameter, sometimes going all the way to closed, but in general, they're breathing and they're pretty large. They're 1 to 2 nanometers, and they can allow all sorts of metabolites if you want. ATP, ATP, glucose, anything like that would be able to come in, ions and water obviously as well. They could possibly thread mRNA, so an mRNA could be passed through. You just have to figure out how to get into the hole, but definitely not transport folded proteins. So just sort of like to be mindful of time, I hope that I've shown you that besides doing in situ structural biology to some extent, we really like this project because it's really allowed us to make discovery. These are projects that we're following right now How do these compartments initially fold? Obviously, how are the proteins imported while nucleases are kept out? And then how does mRNA get exported? I'm going to show you, just to show you the selectivity of this thing, I'm going to show you a fluke but reproducible experiment that we have. In this particular case, fluorescent proteins always stay in the cytoplasm as they should. They're not supposed into the nucleus. For this particular phage, this particular bacteria, eGFP gets imported, but GFP plus and superfolder GFP don't. So that's, it's really hard to imagine what is different about this particular one that has 2 point mutations in a beta barrel that will allow it to come in. And so there's really exquisite selectivity of import. And so, of course, as you can imagine right now, we're chasing the nuclear pore of this thing because we don't think it's chimalin alone. And hopefully next time I see you, I'll be able to tell you a finished story for that. All right, so with that, I just have to say that, around tomography, really using it as a scaffold for what we like to call integrative cell biology, we build many other tools with our collaborators and in my lab that we can then bring together to do all this biological discovery. This project, every time we take a look at a tomogram, we're like, what? Here I want to show you, I hope you can see it on the lower left inside this red circle. There's a pre-assembled genome inside the nucleus, and then you'll see one molecule of DNA going through the portal, getting injected in the phage. But there seems to be the case that the genome gets pre-assembled inside of the nucleus before it gets threaded into the capsid. So you can imagine, like, you would basically knit a ball of make a ball of yarn before you start knitting a sweater. So there has to be a DNA organizing protein that we haven't found, but definitely would look at, et cetera, et cetera. All right, I just want to take 1 minute to tell you about how this project has taken us in very cool directions that are not necessarily cryo-EM, but related. We have a phage class here at UCSD where students go, it's an undergraduate class, students go and hunt for phage and then sequence them, isolate them, sequence them, and a lot of molecular biology. During the pandemic in this class, Amy Pritchard, who's a student in my lab and Joe's, and Gina Lee, who's a fantastic undergrad, help the students basically find all the genomes of what we call now the Chimiliviridae and align them. And the idea was to see, first of all, you can see now why GP105, then it's called GP35 in this genes of whatever. But they found that there's a number of proteins that are conserved across all, across all of the phage, all of this jumbo phage, and they typically come in blocks. But what's stunning about this image, of course, is that phage are normally super tightly packed and every protein is essential. And people have tried to use phage, and repurpose them as tools for other things, but it's really hard to do because you can't really take anything out. They've evolved to be a lean, mean killing machine. But here we can see that there's a lot of sort of like dead weight that we're trying to make sure that that's the case. And it's only about 70 out of 300 to 400 genes that are needed. So, we think of ribosomes as large thing. This is T4 that I told you was a large phage. This is a massive phage with apparently a lot of room for putting like cool things in there. So that really kind of like got us to thinking, is it possible to establish what the minimal phage is, or like the minimal genome that has only all the essential genes, take everything else out, put things in like anti-immune factors to make it, to make it be able to avoid many immune systems in bacteria that people are discovering currently, broaden the host recognition range so that it can infect, different strains. And then for good measure, maybe we'll add antibiotic synthesis so that when the cell explodes, it just releases confetti of antibiotics to others, and then try to train this phage to have an optimized phage pool. And our ultimate goal is really to do individualized bacteriophage therapeutics. So for many, as you know, antibiotic-resistant infections, there's no other way. So people basically get put into hospice. Phage therapy can be used as a compassionate case. And so what we want to do is basically make it an individualized therapy. And Joe and I have assembled a dream team of collaborators. Kevin Corbet, I told you about structural biology. Kit and Joe are bacterial cell biologists, me, cheerleader. John Glass is, as you may know, the guy at the Venture Institute that made a minicell. John can synthesize genomes of almost any size and put him whatever he wants them. So that he's helping us with that. Justin Meyer and Paul Turner are evolutionary biologists that do this training, but also Paul Turner and David Pride represent two phage therapy centers in the United States. IPATH at UCSD, I don't have time to tell you about, but it's a fantastic center with a really good story. So please Google Perfect Predator. And we're very lucky to have had and got funds from the Howard Hughes Medical Institute to try to pursue this. And we want to go all the way to preclinical, which means that we're going to get patient samples. We're studying with Pseudomonas infections for people with cystic fibrosis that they get antibiotic resistant, develop antibiotic resistant pneumonias. I want to say that recently there was a contamination of eye drops in the United States. And I mean, I think in many parts in the world. In the US alone, 6 people died, and UCSD was making a lot of phage because that bacteria was susceptible to one of the phages that I showed you today. So that was really cool. And Paul Turner has a jumbo phage, and it was a jumbo phage, of course. And Paul Turner has a jumbo phage actually in clinical trials right now. All right, with that, I want to thank my fantastic lab. I have the most fun, kind, creative, smart, wonderful lab, and I'm super honored to work with these people. The people that work with the phage nucleus are here highlighted, past and present are here highlighted in blue. And on the left are our collaborators only for the Jumbo Phage project. Naturally, I'd like to thank funding agencies that have believed in our work. And thank you very much for the invitation and for your attention.